Be on the desk at 6, but usually wraps up at like between 6 and 6:30.
Yeah, you can really do make all right.
Yeah.
Yeah, but
I mean, I'm just on the right
Yeah,
there,
right.
so I
Was it
can.
full?
Yeah,
Okay.
I can try to pop back over, clean up.
oh, definitely.
Okay, perfect.
I don't know how to. I'll figure it out once I finish recording.
Yeah.
You have to.
Yeah, I think it does that automatically. I'm gonna say, but not to YouTube. I think Kelsey usually does that.
Right, yeah, that was
Okay.
okay, since that's not rush it off.
Yeah.
You can see attendance like. Yeah, Richard Wade, that's in it, and really depends and. So, you know, like, I think now with, in May, I think it was a sign up, rates. Usually, usually 80%. Since I would say I covered the everything in me. West. And then forward we watched the they do things all the music. But the future features and their I don't know. We got a this would be set up by the termination. Not Oh, yeah. Yeah. I'm excited about the computer. Right, yeah, it was fun. I was working on work and I then opened it, it was like the screen. Right, yeah. That makes me a little bit, yeah. For sure. Can you just tell me I was trying. Sometimes it's like, it's just actually out of charge. And then I've had mine like turned on. It's like, oh, some good charge.
Yeah.
Yeah.
Or sometimes they started updating itself.
Yeah. Yeah. If you really, if I reset it, the sound comes out, but not the or any of it. Like if it could make sense, like a display connector and would change it like link it to an external monitor, yeah, and then I might see something, and then I can just at least move some data over to the USB stick for Thursday, but yeah, but some of these I will deal with it later. I'll record it for the later.
Yeah, I hope it's not a serious problem. Shall we?
Sure. Yeah. All right.
Yeah.
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 You go do the,
You can go ahead.
oh, I think usually,
Oh, the land acknowledgement.
yeah,
Yeah.
and
And
the
then the-
introduction.
Yeah, sure. Okay. So before we get started with our Berkeley Public Library land acknowledgement. So Berkeley Public Library buildings are built on the territory of Huichun, the homeland of the Chochenyo speaking Ohlone people. We acknowledge and honor the Ohlone people who have lived in Berkeley and throughout the East Bay since time immemorial. The library recognizes that Berkeley residents have and continue to benefit from the use and occupation of unceded stolen land. And we pledge to recognize the permanence and the rich contributions that the Ohlone people would continue to bring to the city of Berkeley and throughout the East Bay. We celebrate the strength of the Ohlone people and their culture. Berkeley Public Library is committed to helping create a more just and anti-racist society and furthering the visibility and celebration of Ohlone authors who are specific to the East Bay and Ohlone culture in our collections and programming. This is a living document that was co-written by Ohlone cultural leaders Vincent Medina and Lucas Trevino and the Berkeley Public Library. So welcome to Popping the Science Puzzle. This is a monthly, though might be taking a pause, right? Seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or a topic they find interesting. The organizers are 2 grad students at Berkeley, Kaylin Zhuang and Daniel Bretthauer. This program happens every 3rd Tuesday of the month at 5:30 in the library and on Zoom. If you're interested in checking out their past seminars, you can visit the website and sign up for a listserv to keep in the loop for upcoming seminars. Also check out the YouTube page to view this lecture and see an archive of past lectures.
Great. Thank you so much, Nico, for the introduction. And we're very excited to have our 2 speakers today, Esther and Jenna, to present. And for those joining us on Zoom or watching this on YouTube, feel free to sign up to our mailing list on the QR code. And we also have a host of archive videos from the past seminars, which are very exciting and also great talks from a wide variety of departments on campus. And so for tonight, we have first Esther Kim as our presenter. Esther was born in Korea, but grew up mainly in New Zealand and Australia. She did her PhD in developmental biology in Germany before moving to Berkeley in 2022 to study immunology with Professor Ellen Robbie as a postdoctoral researcher. Outside of the lab, Esther enjoys playing tennis, cooking, and watching films. She organizes the monthly Berkeley Postdoc Association Film Club and is always keen to talk about films and hear others' recommendations. So please take a minute.
Share?
Yes.
Oops. So, hello, everyone. It's a pleasure to be here. Thank you for the opportunity to present. And today I'd like to talk about a category of immune cells called T cells, and hopefully pique your interest in the immune system. Given that we're at the Berkeley Public Library tonight, I thought I could start the talk with the book recommendation. A Gentleman in Moscow by Immortalis. I'm sure there are copies upstairs. And in this book, which is a very good read, by the way, there is a scene where the titular gentleman is at a restaurant and he orders and also the cook. And this is an Italian dish of braised beef that comes with a chunk of bone in the middle. And the soft marrow inside is probably the best part of the dish. And as much as I love this dish, the ossobuco, as a biologist, it's far more common that I think of the bone marrow as a source of blood stem cells and the progeny that they produce. Because the marrow gives you red blood cells that transport oxygen throughout the body, platelets that help with blood clotting, and more importantly for this talk, white blood cells that will defend against infections, foreign bodies, and even cancer cells. And if you look closely inside the bone marrow, what you can appreciate in this image where basically each color represents a different cell type, it's teeming with diversity. And it's important to note that the term white blood cell actually encompasses multiple cell types, each with a specialized function within the immune system. So the cells of the immune system work in a coordinated effort to protect us against invasions, and they can be broadly divided into two categories. So for example, when you have a viral invasion, the first set of cells that will protect you are members of the innate immune system. So this is the first line of defense that operate by this all-purpose alarm system that tries to get rid of the virus. But often the innate immune system alone is insufficient to clear the virus. So you then fall back on the adaptive immune system, and the adaptive immune system is specific for each pathogen, and it has this remarkable property of memory, which means the next time the same virus invades, it will respond much more rapidly and more robustly to protect you from further infection. And therefore, it is the adaptive arm of the immune system that is the basis of vaccinations. And the two major cellular components of the adaptive immune system are the B cells, which produce protective antibodies, as well as T cells, which is my bread and butter, that we study in the lab of Professor Ellen Roby. Now, in terms of how T cells are made, they're made when a group of stem cells from the bone marrow migrate to an organ called the thymus. How do I get rid of that? Well, yeah, so the T cells, yeah, thank you, migrate to an organ called the thymus for maturation. And in fact, actually, the thymus was the last organ in the body whose function was identified. So up until the 60s, people thought, well, it's like a evolutionary mistake, an organ of insignificant function. It's probably just a graveyard where white blood cells go to die. However, a scientist physician by the name Jacques Miller. Do I go? Is it stuck? Sorry. Oh, okay. So Jacques Miller was a physician scientist working in the 60s, and he actually did a series of experiments where he surgically removed the thymus from newborn mice. And what he noticed is that it leads to increased death from infection. And when he followed up this phenomena more closely, what he learned is that actually, when you have no thymus, you have a reduction in the number of white blood cells in the body to protect you against infections. So if you follow this graph here, in the black top black line represents just regular mice that still have their thymi, and as they develop and age, they basically increase gradually the number of white blood cells in the body. At the bottom, the ones that had their thymus removed, you don't get this development of white blood cells. So based on these observations, we discovered that, instead of being a graveyard for white blood cells, it's quite the contrary. The thymus actually makes a group of white blood cells, and these cells became known as T cells. So T is for thymus. And the word thymus itself comes from an ancient Greek word thumos, which means the heart or the seat of your emotions. And as the name hints, it's an organ that is located close to the heart. It overlies the heart and consists of two lobes. And within each lobe, if you look closely, there's regional specialization. So there are, in this particular advanced CT image, there are darker regions and lighter regions. And if you zoom in even closer now using an electron microscopy, what you see is it's full of these round thymocytes closely interacting with these broader fan-shaped cells that are providing a support role for their development. And what's really important to note is that although the thymus is full of these developing T cells known as thymocytes, only about 1% of them will actually leave the thymus into the bloodstream as ready T cells for immune function. And the reason for this is that it's pretty tough to become a T cell. So I mentioned earlier, in the adaptive immune system, the response is pathogen specific. And each T cell is defined by a T cell receptor, a protein on the surface that acts as an ID, as well as an activation switch. And because we have to prepare for all the variety of infectious pathogens that are available in the environment, our body has to produce a huge diversity of T cell receptors. And the way we do this is a special gene shuffling process, and where the genes are shuffled and you get a huge number of different T cell receptor diversity. And just to illustrate how this process works, anyone who enjoys a game of anagrams would know you can shuffle letters together, and that will give you lots of combination of letters, but only some of them will actually have any meaning. In the same way, when we have this gene shuffling event for production of T cell receptors, a lot of them will end up being non-functional. So when and then the support cells in the thymus serve as sort of like a testing ground. So through interaction with the support cells, the thymocytes test out their T cell receptor, and any T cell receptor that is not functional will be eliminated from the system. But that is not the end of the trial and tribulation that the thymocytes have to go through, because there's one other major test that they have to pass. Now, a T cell can be thought of as an elite athlete in a team sport. like LeBron James or Shohei Ohtani. If they're on your side, you feel good. If they're against you, you have reasons to worry. So if a T cell fails to recognize our own healthy cells as sort of being healthy tissues and starts to target it, we end up with what's called autoimmunity, when the T cells of our own body attacks us instead of infected cells. And to illustrate this point, type 1 diabetes is a autoimmune disease where the adaptive immune system attacks the insulin producing cells of our body. Similarly, rheumatoid arthritis is also an autoimmune disease where the T cells attack the joints. And to prevent these sort of autoimmune reactions and to select for self-tolerant thymocytes, we have this pink region in the thymus that we call the medulla. And within the medulla, the support cells are able to provide a sort of a testing menu of the entire body. And the thymocytes, by interacting and trying out this tasting menu, sort of depending on how strongly they're activated or how strongly they respond, if they are self-reactive, they will be eliminated. So basically, the system is selecting for thymocytes with a functional T cell receptor that is also self-tolerant. So by the end of this process, only about 1% of thymocytes will survive and be exported from the thymus to function in the immune system. And this process of development of thymocytes in the thymus is the main subject of study in our lab. And it's the age of sort of sequencing and looking at gene expression. So one recent work that came out of our lab that was done with several collaborators is that we looked at gene expression changes throughout the entirety of T cell development. And this is a visual representation of the gene expression changes, which we call the UMAP. And basically each color represents a distinct stage in the development of the T cells. And what you can appreciate is that within the thymus, there is an assorted mix of cells, assorted mix of immature thymocytes at different stages of development. And this development will culminate in largely two different cell types. One being CD4 helper T cells that have a more sort of coordination regulatory role within the immune system. And then second, the CD8 cytotoxic T cells. And these are the cells that will bind to targets, recognize targets, and actually kill them. But sort of the stage of development that I like to work on in the lab is a much earlier stage of development before they become future CD4, CD8 T cells. And the question that I'm asking broadly is, what traits are important for the development? Are there any factors or components that we can identify in very immature thymocytes that can sort of give us a clue as to their function or their future potential? And sort of the bread and butter methodology I use to answer this question is something called flow cytometry. Basically, this is a technique that allows us to measure proteins in cells. And this is useful because often the combination of proteins in a cell can help us identify them. And in this example, I am measuring the levels of two proteins in a cell, CD4 and CD8. And in this upper left corner, where cells have high expression of CD4, but low expression of CD8, these can be identified as future CD4 helper cells. And conversely, in the lower right-hand corner, where there's high expression of CD8 and lower expression of CD4, these can be identified as future CD8 killer cells. But the cells of interest for me are the cells that are high for both CD4 and CD8, and we call these the double positive thymocytes. And in terms of the specific traits that are important for development, I'm looking at a number of different factors, but for today's talk, I just want to give you a snippet of one work in progress that has to deal with the surface of the cell. So when we measure gene expression or protein expression, these give us very valuable biological insight into the systems. However, if we want to probe function, we always need to think about the way in which proteins or cells actually interact with one another or interact with their environment. And in the case of the thymocytes developing the thymus, I already mentioned that they work together with the support cells. So this is work done in collaboration with Dr. Daniel Lee and Daniel Fletcher's lab, also at UC Berkeley. And we predicted that because two cells have to come close together in the thymus for any sort of development or events to take place, that maybe we can assess the crowdedness of the cells to see whether that impacts development at all. So on the left-hand side here, this is just an example image of a white blood cell. And the cell surface is rich in proteins, different fats, as well as sugars. And in this instance, the black fuzz is all the sugars on the surface. And so given that, white blood, there is some indication that white blood cells have a crowded surface. We applied a special crowding sensor that the Fletcher lab developed, and so we can assess whether the surface of the thymocytes are bare or crowded at different stages of development, and we link that back to their function. And so what we found is that when we applied the crowding sensors and then on top of that, use the flow cytometry to see which cells are at which developmental stage. And we've discovered that the cell surface crowding is dynamic during T-cell development. And whether this has any functional relevance in development, we don't know. It's a work in progress, so we hope to discover more insights into it, but this is all I can tell you about for today. So in summary, I talked about how the bone marrow really is a cradle of the immune system where it generates a lot of stem cells and different types of blood cells that would protect us from pathogens. And in particular, some of the stem cells from the bone marrow are able to migrate to the thymus to become T cells, which are one of the major arms of the pathogen-specific adaptive immune system. And we also saw that a thymocyte becoming a T cell is a very sort of tough journey of a pretty tough journey, because they both need to have a functional T cell receptor as well as a receptor that is self-tolerant. And in terms of ongoing research, I gave you a primer into how cell surface crowding is a dynamic component of developing thymocytes, and this may or may not have functional consequences. And I would just like to end the talk by acknowledging my colleagues and my lab and our support services in our department at the university, because scientific research is always a team effort. And I wouldn't be able to be here today if not for the wonderful team that I work with. And I'd be happy to take any questions.
So I have a question about the timeline. Things you mentioned, thymocytes have to be self-tolerant, and one way to test that is by interacting with the cells in the network.
Yeah.
So how long does it take?
That's a very good question. So it depends. So it varies between cells, but it can take anywhere between like a week to even up to 3 weeks. And so each stage of the process actually takes place in defined regions within the thymus. So I touched on the fact that the thymus has regional differences. And so the thymocytes, although I didn't talk about it, are also able to migrate pretty well. And so if they were just migrating from one place to another, and they can migrate at maybe like 10 microns, give or take, per minute. And if you have like a thymus that's a centimeter, it would only take about a day to traverse through the whole thymus. But given that it can take up to 2 to 3 weeks, there's a lot of pausing, stopping, pausing another test, pausing, interact with another support cell and another test. So it is a pretty long drawn out process. Should I stop share here?
Yes, you can also unplug up too.
And let me see if we can. Thank you.
Thank you so much, Esther. And now I am going to introduce our second speaker, Jenna Manske. Jenna grew up in Michigan and attended the University of Michigan for her undergraduate degree in chemistry. After graduating from undergrad in 2020, Jenna moved to Berkeley and began her PhD in chemistry in the lab of John Hartwig, where she studied ways to make and break bonds with different catalysts. Outside of research, Jenna enjoys taking pottery classes at the Berkeley Art Studio and running. Please take a look. Nope, that's true. Please see your presenters. So if you swap display. Okay, perfect.
Yeah.
Perfect. Okay. Hello, everyone. I'm very excited to talk to you today about some of my work, but also a topic that's very near and dear to my heart, which is catalysis. And so I thought I would start. Yeah. Okay. Oh, perfect. Okay. I thought I would start by kind of introducing some context about my work as a PhD student. So I'm at Berkeley, and this is my research group here. I'm in the Hartwig group, and my lab works generally on developing methods to make molecules more efficiently. So I thought I would show some fun pictures of me in the lab, as well as me working with some of my wonderful colleagues, one of whom presented here a couple months ago, Isaac. I also thought I would show some pictures of kind of what the lab actually looks like when I'm doing the research. And so it's a little chaotic, but generally speaking, the workflow is you put the chemicals up on the shelf, you grab some glassware, kind of very mad scientist-esque. You mix the chemicals together in a safety fume hood, and then you have a chemical reaction and you figure out what you made and whether that's important. And so I thought I would show some context for how we actually develop these catalysts in the research group. Okay, so I thought I would start by just talking about, what is a chemical reaction? And one of the ways you could think about it is kind of looking at the schematic. And so, many of us have played with Legos. You could think about those as the reactants or starting materials. And so when you have this arrow, that's the reaction happening in the forward direction. And so now you have these products. Once the chemical reaction has happened, we can think about those as the Legos kind of stacked in a different conformation. And so chemical reactions rearrange the atoms, but you're not actually changing the atoms. So again, you kind of can go backwards and get the Lego blocks out. You haven't changed anything fundamental, but you have a product that's different than the starting material. And so I would thought I would show the periodic table. So here are the elements or atoms. And so the letters don't change, but the arrangement of the letters does. And I thought I would show that with some chemical structures on the board. And so the red line is just showing a chemical bond. And so you can see that the connection between the letters is the chemical bond. And although you've changed, right, you've taken your starting materials and now you have some products, you've changed the arrangement of the letters, but at the end, you still have the same letters in your product that you had in your starting material. And so again, chemical reactions are making or breaking bonds, but you stay with the same elements. Okay, so I thought I would also kind of tie it back to a chemical reaction that you may have done at your own house. So many people maybe have mixed vinegar and baking soda and observed some outcome. And so what that actually is, what's happening on a chemical level is that you're taking this bicarbonate or baking soda, you're mixing it with vinegar or acetic acid. And what's happening is you're forming this carbon dioxide, water, and then acetate. And by doing this, the carbon dioxide, which is a gas, forms these bubbles. And that's the very cool volcano eruption effect that you may have observed. And so all you're observing is essentially a chemical reaction where you're taking your starting materials and your products and you get the volcanic eruption. But another example is baking. And so you kind of might have a very similar set of starting materials and that you still have bicarbonate, but now you have flour and eggs and sugar. And you set up this chemical reaction the same way we do in the lab, right? You mix everything together in a bowl, you put it in an oven, just like we might heat something with a heating bath. And then you get a product, which might look like this beautiful cake, but the way that the cake kind of forms in such a fluffy, delicious way is because, again, similar to the volcano that we've had before, you're also forming carbon dioxide, which forms bubbles and then gives your cake the very light, fluffy appearance that you want. And it's a chemical reaction. Okay, so back to kind of explaining about catalysts, right? So now we maybe are comfortable with chemical reactions, but how are they happening? How can catalysts help us, etc.? So you might want to think about your starting materials, right? The chemicals on the left side of the arrow as sitting in a valley. So just like you may be sitting with some hills, this chemical has a decision about what to do. Does it want to stay in the valley? Does it want to try and traverse one of these mountains or hills? And which hill is it going to pick? Is it going to try and go for the one that's the lowest hill to climb or the highest hill to climb, etc.? Starting material in a valley. But just like I said, to actually react, right, to undergo a chemical reaction, the starting material does have to climb one of these hills. It has to climb into the next valley to form its products. And this requires energy, just like it requires a lot of energy for you to climb one of these mountains yourself, right? And so, you know, it's nice to think about the hills and valleys, and that's kind of what's really happening from a chemical sense. It's just much more complicated. In reality, the chemicals are sitting in this multidimensional landscape. There's many different local peaks, many different local valleys, and they have a decision about where to go based on the energetics and how much energy they have. But we like to kind of simplify the very complex landscape into a 2D depiction that helps us think about how our chemicals are going to react in the flask. And so this two-dimensional landscape looks a little bit less complex than the multi-dimensional landscape, but essentially it's the same thing, right? You have your reactants, they're sitting in a valley, they have to traverse a pathway or a hill, right, to get to the products. And the actual amount of energy that it takes to climb the hill is called the activation energy. Right, so just like you think about yourself, you need the activation energy to climb a hill, to climb a mountain. The chemicals need that as well. And so this is just another depiction, right? You have this activation energy. You can also think about yourself walking up the hill, but imagine if you had to push a rock up the hill, right? It takes a lot of energy. And again, that's the activation energy. So the amount of energy it takes for the starting materials to go over the hill to form products. And so that's what's happening during a chemical reaction. Hills are very hard to climb. And so chemists have thought about, chemists as well as just generally the world has thought about how we can figure out how to lower the energy to make it easier for these chemicals to form products. And so that's what a catalyst is. So again, you've got your starting materials, you've got your products, also have this term catalyst. And a catalyst is not going to be used in a chemical reaction, so it'll be there at the end of the reaction. You're not actually using it, right? You don't see a green Lego in the products. It can be used over and over again, so it's not going to be consumed. But if we go back to our two-dimensional energy landscape, what the catalyst is doing is just lowering the activation energy. So it's lowering the barrier to make it easier for the chemicals for the starting materials to form products. And it does that in a variety of different ways. But how does it actually help? So it's kind of a way that you can think about this where it might be relevant to you. If you think about hydrogen peroxide sitting in your cabinet, sitting in a bottle, right? It doesn't just decompose on its own. So if you don't have a catalyst, your hydrogen peroxide isn't going to form oxygen and water in the bottle. Or if it does, it's going to take many years, many months. There's an expiration of the date on the bottle, but it's not, a couple weeks. It's definitely a long time. And so the reaction has a very high activation energy if you don't add a catalyst. So you could think about that as a map type, right? It's a really, really hard barrier for the hydrogen peroxide starting material to break down and form products without a catalyst. But if anyone has ever had a cut and put hydrogen peroxide on it, you'll notice that it fizzes really, really quickly, right? So you're breaking down the hydrogen peroxide into oxygen and water very quickly. And the reason is that your skin actually has, or the inside of the cut, has a bunch of catalase enzyme, which serves as a catalyst to break down the hydrogen peroxide into water and oxygen. So now the reaction has a much lower barrier and it can happen in a number of seconds. So you could think about that as now a gentle hill for these molecules to go over to form the products. And so I thought I would now talk about a variety of different catalysts. So in my lab, I work on two different types. I work on, biocatalysts or catalysts that are found in biology. I also work on metal catalysts, and I'll talk about those next. So I thought I would give two examples. So the first is a biocatalyst, also known as an enzyme, and it's a polypeptide, so made of a bunch of amino acids. It's like a protein, like you might take as a supplement, like proteins. And these biocatalysts serve by taking the starting materials or substrates and bringing them in very close proximity to each other, as well as changing the chemical environment of the substrates to now lower that barrier. So you can think about them as taking starting materials, bringing them very close together, and then you can form the product, again, lowering that barrier, in this case by proximity. So where's an example? Can anyone think of an example, I guess, that might be related to milk? Okay, cool. One is an example of lactase. And so lactase, when you drink milk or take a dairy product, you have an enzyme that breaks down lactose into galactose and glucose. And so this is a very hardworking enzyme or biocatalyst, right? It does its job very well. Takes, again, this is just your simplified picture, and then your chemical structure is here with the lactose being broken down by lactase. And again, if you have lactase, if your body produces it, you're in good shape. You can break this down. You don't have any challenges. However, people that are lactose intolerant now don't make this catalyst. And so now the barrier is really high for this lactose to break down into glucose and lactose. And so what they have to do is actually eat lactase enzyme so that they have the catalyst inside of them, so that they can break down lactose, and they don't have to rely on bacteria to do it, which can cause painful experiences. And so I also just thought I would highlight a very prominent scientist in the field who's really been driving innovation for biocatalysts. And that's Frances Arnold, who won the Nobel Prize in 2018. for her work on directed evolution, which this is a very complex schematic, but essentially Frances Arnold came up with a workflow so that you could very rapidly change the types of chemistry that can be done by biochemicals. So she really expanded the way that people think about using enzymes to do chemistry. Okay, so I also thought I would highlight types of catalysts that are metals. And so this is going to be a little bit of a complicated cycle, but I thought I would just highlight that in this case, we're taking palladium, which is a metal, and we're using it as a catalyst. And so the way that this works is that you take a starting material, and this is going to be drawn in a catalytic cycle or a catalytic circle. And so that's just showing you again that the palladium starting catalyst ends up as the same catalyst at the end. So you're not consuming the palladium, you're just using it to bring your starting materials together to form products. So you have palladium, it interacts with the starting material, which is called an aryl chloride. Now you have a palladium complex that seems like it's broken the bond between the aryl chloride, the carbon and the chloride. And then you bring in another starting material that can also interact with the palladium. And so now you have an even more complex palladium catalyst, but this catalyst has now brought these two starting materials together, so they're in close proximity, almost like what the enzyme did for the biocatalyst. And now it can do a fancy term called reductive elimination, where it brings the two components together and forms this product. But again, the cycle kind of connects and you end up with the same catalyst. So this is the type of catalytic cycle or circle that people in my research group, as well as other research groups, think a lot about how we can change the catalyst to improve the chemistry, how we can tune starting materials and products. And so that's kind of how the metal catalysts work and how we like to draw them. So I thought I would, think about another catalyst that people may be familiar with. So does anyone know what catalyst might be in a car? With rhodium. Okay, cool. Well, if anyone's ever thought about catalytic converters, they're just metal catalysts that take engine emissions, which are kind of toxic, dangerous gases that we don't want in the environment, we don't want to be breathing in. And the catalytic converter is just made of metal heterogeneous catalysts. And they do their chemistry, lower the barrier for the chemistry to happen, so that your actual tailpipe emission are benign components such as water, carbon dioxide, and nitrogen that are not going to be as dangerous for your health. And so this is something we've all interacted with if you've interacted with a car. The catalytic converters are out there to help the environment and you. Okay, so then I thought I would circle back, talked about catalysts. How does this relate to my research? And so first I wanted to show two chemicals that maybe you might be familiar with, Advil and Tylenol. So here are the chemical structures for those two molecules. And, from a chemist's perspective, they're not as complex as other structures, but they're still pretty complicated. There's a lot of different lines, a lot of different bonds that are connecting them and might be challenging to make. But if you look at an even crazier molecule, such as Paxlovid, which was really important for COVID-19, it's a much more complicated molecule. Now there's tons of different bonds, they're all very complicated with even more elements involved. And so it's important for kind of people, the way that we like to think about it in our lab is we like to help build tools or catalysts that can help make these different bonds and complex molecules so that later down the road, people at companies can actually make drugs using our methods or tools for catalysts in a more facile way. And so I thought I would just highlight two ongoing research projects. And so one of them is using a biocatalyst, right, an enzyme, to convert a carbon-hydrogen bond into a carbon-chlorine bond. And so that's involved a lot of development of this biocatalyst, changing it to interact with the substrate in a more efficient way so that we can accomplish this challenging transformation. Additionally, I also work on metal catalysts, so now using an iridium complex that undergoes that similar catalytic cycle as I showed before, but now using the starting material that has, again, a carbon-hydrogen bond and taking this catalyst to now install a carbon-boron bond, which can be useful for other downstream transformations. So again, developing a catalyst, thinking about that catalytic cycle, how we can improve it to make this reaction, again, more facile. Okay, so in summary, I've talked a little bit about some chemical reactions that you may not have known were so essential, but are very fundamental to everyday processes. I've also talked about, how chemical reactions require energy for the molecules to overcome this barrier of activation energy. And then how catalysts work to lower the barrier to allow the chemical reactions to happen faster. And then how in the lab at Berkeley, we're developing new metal catalysts as well as enzymes to accomplish challenging transformations that could be useful far down the road for making drugs. And with that, I have lots of people to thank. I'd like to thank my wonderful lab, my wonderful undergrads, everyone I have the chance to work with at Berkeley, as well as funding, and all of you for your attention and organisms. And I'm happy to take questions.
What are the criteria for evaluating if it's good?
Oh, that's a great question. Generally speaking, if it can work fast and not like deactivate. So one of the biggest problems is catalysts are really good at the beginning and then they lose steam and don't turn over. And that can be through a variety of different reasons, but working on really robust catalysts. So if it doesn't decompose over time, that can be really important. If you can reuse it, that's even better. So if you can resubject it and have it still work, but generally having fast catalysts that really lower the barrier a lot can be the most useful, but great question.
What were some strategies to find a good catalyst?
Yeah, that's a great question. So if I go back to the picture of the catalyst, that one of the ones that I'm looking on. So normally you take this chemical structure and you have kind of an idea of what catalyst generally might work. So I want it to kind of look like this and kind of look like this. And then you just change the chemical structure a little bit, bit by bit. So you maybe delete this part here or add a bigger part there, or change this part here. So you just kind of fine tune the catalyst. And so to make this catalyst, I had to make maybe 15 other different catalysts that looked similar, but were slightly different. Yeah, it's a great question. So kind of fine tuning of different properties to try and get changes.
Do you use AI to help you make predictions about what structures would lead to what, sort of whether improve or dampen catalytic function?
That is an excellent question. I personally, in my research, that is absolutely ongoing, and my research group does. The challenge is that the chemical literature actually has a lot of things that are inconsistent. And so often you have to regenerate large data sets so that you can then use AI to kind of pick through different pieces, but that's absolutely ongoing, right? Because you can make 30 different ligands, test them under controlled reaction conditions, and then build a model. So yes.
So you had mentioned that to find like a good catalyst, you compare what you have and what you want it to be. So for that, is the catalyst sort of like the missing pieces, or is it sometimes like something completely different?
Like what the beginning catalyst did versus the end catalyst. Yeah, it can change quite a bit. The nice thing is for a lot of different projects in the lab, we have a large library of kind of random catalysts that kind of all do different things. And so when you think about, a new transformation you might want to do, you want to take this carbon hydrogen and turn it into a carbon chlorine or carbon fluorine. You can kind of screen, not randomly, semi-randomly, get a starting point, and then you start to build out. But yeah, it can look one way and then change to be something pretty different at the end. Yeah, great question.
Awesome, thank you.
Awesome, thank you. And
Awesome. Okay, let's give a round of applause to both of our speakers again. And the last slide that I have for everyone is if you're interested in today's talk and want to hear it again, or if you like to hear more, please join our mailing list by scanning this QR code. We will be skipping June. We're taking a break in June and we'll come back in July. The next seminar is Tuesday, July 15th at 5:30, and we'll be at the West Branch. So not the Central Branch location, but we'll be at the West Branch, and same Zoom link if you want to join on Zoom. And that is all I have for today. Thank you all. Thanks to the speakers, and we'll see you in July. Oops.